Hello, everyone. Thank you so much for the opportunity to present here today. This is our presentation on our project, the development of lipopeptides as bioactives for drug-resistant fungi. I'm Netra.
I'm Nishika.
I'm Maya.
And our supervisor is Mr. Amadi. So let's start with a brief agenda of the content that we're going to be covering today. We're going to be starting with the background of our project, and then our research objectives, and then our methods and expectations, current progress, results and data analysis, conclusions and next steps, and we're going to finish off with a Q&A opportunity for the audience.
So starting off with our background. So here we have a few key definitions that are pretty crucial to our project. First off is drug-resistant fungi, which are fungal strains that have developed a resistance against certain drugs due to constant use. Second, we have lipopeptides, which are a lipid attached to a peptide. And lastly, we have bioactives, which are compounds that affect living organisms.
Currently, there is a big problem for the global banana production industry. That is, the fungus FOXTR4 causes the Panama disease or Fusarium wilt that is very deadly to Cavendish bananas. And these bananas make up 95% of global banana exports. Unfortunately, TR4 is highly resistant to current agricultural grade fungicides. Therefore, we are testing lipopeptides on drug-resistant fungi as bioactives to inhibit the Panama disease.
So here are some current treatments for TR4. And the reason why we are investigating lipopeptides is because there are big limitations that comes with these treatments, mainly biopesticides and fungicides. And because of increasing pesticide and fungicide resistance, these treatments aren't that reliable as of recent. And here we've listed B. subtilis, which kills TR4 by breaking down pathogen cell membranes, Vacetyl aluminum, which restrains spore germination, and fluopyrum, which stops TR4 cellular respiration by stopping the ETC.
Now let's talk about our research objectives, which are pretty much the guiding questions that we ask ourselves throughout our project. So the central question, and really the first question that we ask ourselves is it possible for us to synthesize a lipopeptide capable of inhibiting TR4? And leading up to this question, we need to understand what target in TR4 our lipopeptide will target. So our treatment could work by denaturing vital proteins to the development of the disease, or also vital DNA sequences. So after you've actually created our lipopeptide treatment, we need to determine the competitive advantages of our lipopeptide treatment in comparison to other existing fungicides in the field. For example, our lipopeptide treatment might be more cheaper or more efficient to produce than other existing treatments. Other than that, we need to think about the possible effects that our a treatment will have on the plants that we are trying to protect.
So next up is methods and expectations. Okay, so our first method that we're going to talk about is the antifungal susceptibility test, otherwise known as the AST. So what we did is we used PDA plates and we cultured yeast, and then we tested the efficacy of various fungicides on the yeast to see how well they affected its growth. So how we did this is by splitting up a PDA plate into 4 quadrants, and then swabbing the plate with yeast, and then and then placing a bit of each, a small amount of each fungicide in one quadrant. So what this does is it serves as a positive experimental control to compare it to our lipopeptide in the future. The fungicides we have tested so far are CTAB, eugenol, methyl eugenol, copper sulfate, hydrogen peroxide, and baking soda.
Next, we use molecular docking to test the binding affinity of multiple molecules to our target protein, which is FOC-ECM33. This controls the reproductivity and virulence of FOXTR4. This is done
louder, please. Louder.
This is done using the softwares AutoDocVina and BioVia. We are looking for a binding affinity of -8 or lower, and these molecules have a binding affinity of around -4.
Another big part of our project is peptide synthesis for the peptide component in the lipopeptide. So first we'll have to print in the synthesizer by pre-swelling resin to help with the bonding and loading in the necessary amount of amino acids. We'll run our reactions, and after that we'll be taking our resin and peptide off, cleave the resin off using solutions made of TFA, water, and TAS, and we'll be rotovapping the more harmful solvents after that. But to confirm that there was a peptide printed in the first place, we do need to run a few processes such as the Bradford assay to determine the concentration, or LCMS to look for a molecular weight similar to our peptide, or gel electrophoresis. But we found this technique to be a little less effective just because we are printing more low molecular weight peptides.
So the CSPPS protocol is kind of a supplementary to our project, as it aims to increase the efficiency of our testing methods, as we could theoretically print an exponentially larger quantity of sequences at a time in a singular reaction vessel. So the idea is that there's going to be kind of a soup of amino acids that will randomly attach to a growing peptide sequence that is suspended on the resin. And so again, with this, a large number of sequences can be created at a single amount of time, which makes printing really fast, and it really speeds up the timing for research and development. However, this method does have some issues. For example, loading efficiency. We're pretty much limited to a 0.25 millimole amount of the initial weighing resin, which has a limited amount of amino acids, and therefore pretty much determines the number of peptide sequences that can be created. Other issues include separating peptides based on sequences and also identifying them based on sequences. Here is an image of the traditional SPPS protocol. So as you can see on the right, amino acids pretty much on the peptide synthesizer print from C terminus to N terminus. And so you start off with your weighing resin, and then you add in a single amino acid at a time, which is determined by the inputs that you put into the machine. And it's really not random at all. And so the output is multiple permutations of a singular peptide sequence. And so the number of peptides printed correlate to the number of the C terminal amino acids that are bound to the weighing resin. And the whole peptide synthesis time takes
3 weeks
hours per amino acid.
Finish it now.
Now let's go
Finish all
over
the 3 weeks.
Okay, and then you can see this image
Can you
highlights
finish it first?
the SPPS protocol. So again, it also prints from C
You want to watch
terminus
this for
to
5
N
minutes?
terminus, and the number of peptides printed correlate to the number of amino acids that are bound to the initial weighing resin. However, in the middle, you can see that the amino acid synthesis itself is pretty random, as not all of the amino acids are placed separately. So it's like a semi-random system of amino acids binding to the growing peptide sequences.
Okay, so our last method is SPPS capping. So this is a method that we haven't utilized yet, but our plan is to do so in the next few weeks. So we plan to link lipids and NHS biotin to the N-terminus amino acid of the peptides we print. The N-terminus is the amino acid that is printed last. So adding this altered amino acid to the peptide synthesizer would create a lipopeptide. The purpose of the biotin is so that we can efficiently isolate the peptide. Using magnetic streptavidin beads that are attracted to the biotin, we would be able to do so. So the purpose of this is to diminish the need for extra purifying steps, and which would streamline our research.
With that being said, here is the current progress we have made. We have tested the binding affinity of the fatty acids lauric acid and propyl gallate to FOC EMC33 using molecular docking. Out of around 20 molecules we have tested so far, lauric acid and propyl have the highest binding affinity to our target protein.
So here are the peptides that we've printed as of recent and the ones that we've been investigating more. First, we have DVDG, which is the sequence of a peptide that signals for caspase protease, which initiates apoptosis. We printed it and we confirmed its identity and its concentration in the Bradford assay. However, we could use more efficiency and because it is a low molecular weight. So that's why we printed DVDG squared to make a longer sequence to make it easier to detect and identify.
And so for disk diffusion tests, what we do is we take an empty agar plate and we swab some pre-incubated yeast on there. And then we split the plate up into 4 quadrants so we can test the efficiency of 3 different fungicides at a time, and also include a control group to make sure that if the yeast is killed, it's not due to uncontrollable factors. We also repeat the experiment multiple times using 4 to 6 plates for the same fungicides each time we perform the antifungal susceptibility test. So we found most of our fungicides on papers that typically deal with a fungi, for example, TR4 like CTAB. However, we've also tested home solutions like baking soda.
So moving on to results and data analysis. So as mentioned earlier, when looking at our molecular docking tests, we are looking for a binding affinity of -8 or lower. None of our tests thus far have reached this number, but the 3 molecules that have gotten the best binding affinity are lauric acid, triethanolamine, and propyl. We will be running more tests in the future using Autodoc V9.
And using mass spectrometry to verify our peptide DEBDG, and we use mass spectrometry to verify our peptides DEBDG and DEBDG squared. For DEBDG, we found a peak that is approximately 217 Daltons smaller than expected, which is around 1 to 2 amino acids that could have been cut off somehow during peptide synthesis. And for DEBDG squared, the sum of the 2 peaks molecular weight, 372 and 763 Daltons, is approximately 162 Daltons over the estimated molecular weight of DEBDG squared. This could be because an extra valine was printed during peptide synthesis. Also, the peptide sequence could have been cut off during synthesis, explain why they appear as 2 smaller peaks rather than 1 larger peak. Also, currently, we are comparing the estimated weight of DEBDG squared to our peptide sequence with amino acids, but with different charges.
So far, here are all the peptides we have printed, starting with our practice, Alaloo, then DVDG, DVDG squared, and now surfactant, which is involved with our supplementary project on combinatorial peptide synthesis, where we plan to print surfactant, the first 6 amino acids, but after that, we plan to utilize the peptide library technique to make a bigger peptide through the peptide library, and eventually attach it to a lipid because surfactant will be part of our lipopeptide.
Yeah, but I didn't get. So for the disk diffusion tests, we know how efficient each fungicide is based on the area that's cleared around the paper plate that's placed at the center of each quadrant, as this is where we place our fungicides. So with this information, we've determined that copper sulfate and eugenol are the most effective fungicides that we've tested in so far. And so this means that in our tests with our actual lipopeptide treatments, we're likely to use these 2 as positive controls.
Lastly, we have our conclusions. So we know our peptide synthesis trials have been successful in printing a peptide, but we have not verified that they printed the exact sequence we want. With respect to AST and disk diffusion tests, we know that eugenol, methyl-eugenol, and copper sulfate are effective fungicides that we can use as positive controls. The target protein we have identified is FOC ECM33, and we'll continue to run molecular docking simulations using this target protein.
So here are next steps for this project now that all we've learned. We want to continue improving our technique in peptide synthesis, especially in verifying the identity, because we could see some improvement in that. It hasn't exactly matched yet, especially with LCMS. We also want to work on combinatorial peptide synthesis. And then we also will be developing lipopeptides, especially with the help of combinatorial peptide synthesis, and working with our sister group MP7, which is working on the lipid component of the lipopeptide project. We'll also continue molecular docking to find potential lipids to bind.
And that concludes our presentation. Here are the sources we used. Also, we would like to thank ASDRP for funding our research, Mr. Yamamoto and Dr. New for instrument training, and Sophia Zhang for her contributions. Lastly, we would like to thank Mr. Amadi for mentoring us.
Thank you so
That
much
brings us to the Q&A segment. If anyone has any questions, please raise your hand or type your question in the chat. Thank you.